Good morning, dear students. This is your teacher Neela Mehlawat, and today we will start the chapter 11. Dear students, this chapter is very, very important, and we know this chapter is biotechnology, the principles and the various processes. And in this particular chapter, we'll be talking about the principles of biotechnology, what are the various tools that we utilize for producing the recombinant DNA, the processes of recombinant DNA technology. So we will deal with 3 aspects in this particular chapter. Now, before we go further, dear students, we must understand what exactly is biotechnology. Just split this word biotechnology. Bio and technology. Just see this, what this word actually means. Biotechnology. What do you understand from the word bio? Bio word actually deals with life. And technology means the various techniques, the various methods. So we can define biotechnology as a science which deals with the techniques of using live organisms, of using enzymes from one organism into another to produce various products and the processes that are useful for human beings. Means we have to utilize the technology to produce certain objects, certain process, certain products that are useful to the human organisms. That exactly what is biotechnology. Now this particular biotechnology, it deals with various aspects. Like first of all, it deals with microbes mediated processes. Microbe mediated process means the various microorganisms which are used to produce useful objects, useful products for human beings, like making curd. As we all know, dear students, while making curd, which organism is active? Lab. Very good. Lactobacillus acid producing bacteria, isn't it? While producing bread, we also require some microorganism. While producing wine, we also require some organisms. So that is why we name it as microbe-mediated processes. The processes where microbe is required. The second aspect deals with in vitro fertilization, right? Which leads to the production of a test tube baby. We can also use biotechnology for synthesizing a gene, a desirable gene, and using that gene to cure some disease or to produce some product which is useful for the human beings. Then we can also use biotechnology to produce the DNA vaccines. Isn't it, dear students? Nowadays we know that in our vicinity, the COVID-19 is creating havoc. And lots of scientists are working on the aspect to develop a DNA vaccine for this particular virus. So in this particular field, biotechnology plays a very important role. Then for correcting a defective gene. Suppose if there is any gene which is not performing its desired function. Suppose there is a gene A and this gene A has to produce a protein. But because this gene is not able to function, it is not functioning well. This gene is not functioning well. Because of its non-functionality, the protein is not being made. So biotechnology can also correct this gene. So that it can start producing the protein and the normal body functions can be restored inside the human beings. So we have 5 aspects in the biotechnology. Now, as per the European Federation of Biotechnology, we have done a general definition of biotechnology. As per the European Federation of Biotechnology, it defines it as the integration of natural science in organisms. Right? This European Federation defines biotechnology as the integration of natural science in the organisms, cells, and parts thereof, and molecular analogues for products and services. means we are integrating the natural science and the organisms or the parts of the various organisms or the molecular substances for the production of various products and services for the human beings. Now coming to the principles of biotechnology. Among many, there are two core techniques that enable the birth of modern technology. आज हम जिस टेक्नोलॉजी की बात कर रहे हैं वो टेक्नोलॉजी दो मेन टेक्नोलॉजीज को क्लब करके बनाई गई है. द वेरी फर्स्ट इज द जेनेटिक इंजीनियरिंग. ओके एवरीवन, द वेरी फर्स्ट टेक्निक इज रेफर्ड टू एज जेनेटिक इंजीनियरिंग. नाउ दिस टेक्निक बेसिकली it is a technique in which the genetic material, whether it is a DNA or it is an RNA, like in case of human beings, we have DNA as a genetic material. But in some viruses, in certain organisms, the RNA also acts as a genetic material. So genetic material, it is chemically altered. Altered means it is changed. So in genetic engineering, the DNA or RNA, it is altered, and the altered DNA is then introduced into the host. Okay, and that brings about changes in the phenotype, in the morphology of that particular organism. The second technology, which is called as bioprocess engineering. Okay, the first that we have is a genetic engineering, and the second that we have is bioprocess engineering. What do we call second one? Bioprocess engineering. Okay, bioprocess engineering. Now, other name for this is maintenance of sterile conditions. We can also call it as bioprocess engineering, or we can call it as maintenance of sterile ambience. Means the maintenance of and contamination-free environment. It is free from all microbes. Now, this particular environment, it is necessary in chemical engineering for growing desired microbes or eukaryotic cells in large quantities. Suppose if you want to grow a particular organism, if you want a cell should multiply at a faster rate, we should provide a nutrient medium to the cell. And when we provide a nutrient medium to the cell, it should be free from contamination. Okay, it should be free from all contaminations. And then only we can use those cells or those rapidly multiply organisms to manufacture the antibiotics, vaccines, or producing the various enzymes. So our biotechnology basically has two main techniques. One is genetic engineering and second is bioprocess engineering. Now coming to the principles further, there are basically 3 steps involved. If you want to genetically modify an organism, अगर हम चाहते हैं कि एक organism का DNA modify हो जाए, तो there are 3 steps that will be done used in this particular method. First thing is that we will identify the DNA which is having the desirable genes. Okay. Now, first of you, as I told you, first of all, we should identify a desirable gene. Desirable gene means, suppose this is an organism, right? Suppose there is an organism, and it is its DNA, and there is one gene present in this particular section, right? That makes this organism resistant to diseases. ठीक है? हम एक ऑर्गेनिज्म की बात कर रहे हैं. उस ऑर्गेनिज्म के अंदर उसका डीएनए मटेरियल है. उस डीएनए मटेरियल में एक जीन ऐसी है जो उसे बीमारियों से रेजिस्टेंस प्रोवाइड करती है, जिससे कि वो आसानी से बीमार नहीं होता. सो वी हैव टू आइडेंटिफाई दिस जीन. Okay, if we are able to extract this gene, we are able to isolate that gene, and we can introduce this gene into other organisms. So other organisms will also develop resistance to a particular disease. So first of all, we need to identify the desirable gene. तो सबसे पहले हमें desirable gene को identify करना है. Okay. Now, up in this technology, in biotechnology, basically, before biotechnology, we used to have traditional hybridization techniques, and these traditional hybridization techniques, they used to multiply when we used to have a DNA, and when we use these particular techniques, and so what should have happened when we are allowing the cells to multiply? So in addition to you can say desirable genes, undesirable genes also multiplied. तो जब हम ट्रेडिशनल मेथड्स यूज़ करते हैं उस केस में जो डिज़ायरेबल जीन है उसके साथ साथ हमारी अनडिज़ायरेबल जीन्स भी मल्टीप्लाई हो जाती हैं, दे ऑल्सो इंक्रीज इन नंबर. लेकिन जेनेटिक इंजीनियरिंग जो हमारी टेक्निक है इसमें सिर्फ हम डिज़ायरेबल जीन्स को मल्टीप्लाई करा सकते हैं. दिस इज़ द एडवांटेज ऑफ जेनेटिक इंजीनियरिंग. हम जिस जीन को सिलेक्ट करते हैं सिर्फ वही जीन मल्टीप्लाई करेगी. ओनली दैट जीन विल इंक्रीज इन नंबर, दैट विल इंक्रीज, and that will be introduced into another organism. Second is, now we have identified that particular gene. Now we will extract that gene and we will insert into another host. हम उस जीन को एक्सट्रैक्ट करेंगे जो जीन हमने आइडेंटिफाई की है और उसे हम किसी होस्ट के अंदर इंट्रोड्यूस करेंगे. ओके, नाउ अ पीस व्हेन इट इज़ इंट्रोड्यूस इनटू अनदर ऑर्गेनिज्म, जब हमने उस जीन को, उस डीएनए सेगमेंट को किसी और ऑर्गेनिज्म में इंट्रोड्यूस किया है, तो दिस डीएनए कैन नॉट मल्टीप्लाई ऑन इट्स ओन. ये जो डीएनए है ये अपने आप मल्टीप्लाई नहीं कर सकता, बट इट हैज़ टू बी इंटीग्रेटेड विद द डीएनए ऑफ द होस्ट. ठीक है, ये डीएनए के होस्ट के साथ इंटीग्रेट होगा तभी वो मल्टीप्लाई कर सकता है. ओके, सो जो डीएनए हमने इंसर्ट किया है होस्ट के अंदर, फर्स्ट इट हैज़ टू बी इंटीग्रेटेड विद द जीनोम ऑफ द होस्ट, then only it will multiply and it is inherited from one organism to its offsprings. The third is maintenance of introduced DNA in the host, means the host will continuously keep on multiplying, and along with its DNA the new DNA or the new gene will also multiply, and then that particular gene will be transferred from the parent to its children or to its offsprings. So we basically this technology involves 3 steps: identification of DNA with desirable genes, introduction of the identified DNA into the host, and maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Now to talk about initially the first recombinant DNA. Now first such DNA, okay, this first recombinant DNA was emerged, or you can say was isolated by Stanley Cohen and Herbert Boyer. You can see in the picture, this is Stanley Cohen and Herbert Boyer. They in 1972, they isolated the antibiotic resistance gene. Okay, they isolated an antibiotic resistant gene by cutting a DNA piece from the plasmid. Okay, and the plasmid was of Salmonella. You can easily see this organism here in the picture. This is a bacteria. So that acted as a plasmid. Okay, that acted as a plasmid. Clear? Plasmid means the organism, the host in which the DNA was introduced and then it started multiplying. So they were the two who identified the first gene, or you can say they were the ones who first produced the recombinant DNA. And thus there was a birth of recombinant DNA technology. और इसी कॉन्सेप्ट के साथ, इस एक्सपेरिमेंट के साथ ही recombinant DNA की शुरुआत हुई. Now recombinant DNA technology basically, students, it involves joining of 2 DNA molecules from 2 different species. Yani suppose there is an organism A, there is an organism B. So there will be DNA from these 2 organisms, right? Like suppose A has this DNA, B has this DNA. Okay, so portion of this DNA will be introduced into the another. Okay, and it will join with the DNA of the B. So thus it involves joining of DNA of two different species that are inserted into the host to produce a new genetic combination. So when the DNA of A, some portion or some genes of A were introduced into B, it will bring about the new genetic combinations. Let's talk about the concept further. Now suppose this is our foreign DNA, okay, and this is the vector DNA or the plasmid, right? This is a foreign DNA, right? हमने एक DNA आइडेंटिफाई किया है जिसके अंदर एक जीन ऑफ इंटरेस्ट मौजूद है, ठीक है. यहाँ हम जीन ऑफ इंटरेस्ट को रेड कलर से इंडिकेट कर ले. सपोज करिए ये जो यहाँ पर एक जीन मौजूद है जो हमें चाहिए, ओके, एंड वी आर यूजिंग अ प्लास्मिड. सो नाउ आर रिस्ट्रिक्शन एंजाइम्स, द एंजाइम्स दे विल कट बोथ द DNA. सो देखिए यहाँ पर हमने प्लास्मिड के DNA को भी कट कर दिया है और हमने फॉरेन DNA को भी कट कर दिया है. एंड दिस इज़ द पीस दैट हैज़ टू बी इंसर्टेड इनटू दिस ऑर्गेनिज्म. ये DNA पीस जो है ये आपका प्लास्मिड के अंदर इंसर्ट करना है, एंड दिस विल बी इंसर्टेड बाय एन एंजाइम विच इज कॉल्ड DNA ligase. Okay, DNA ligase, it will join both the DNAs. So now we have inserted the DNA. Just you can see this is the portion that has come from here. अब आपका जो प्लास्मिड है उसके अंदर दोनों DNA मौजूद हैं. एक उसका अपना और दूसरा उसके अंदर फॉरेन DNA इंट्रोड्यूस कर दिया गया है. Now this DNA molecule will now be called as recombinant DNA molecule. What do we call this? Recombinant DNA molecule. Okay. Now this recombinant DNA molecule, now it will multiply in the cell. अब ये जो है सेल के अंदर मल्टीप्लाई करेगा. इसको हमने E. coli के अंदर इंट्रोड्यूस कर दिया है. Okay, we have introduced this recombinant DNA into the E. coli, which acts as a cloning host. Now when the E. coli will multiply, this recombinant DNA will also multiply and it will be transferred to the future organisms. तो recombinant DNA को पहले हमने दो DNAs को insert किया, उन्हें एक बनाया, उसको हमने कहा recombinant DNA. Now this recombinant DNA we have introduced into a cloning host. Now this host, when it will duplicate its DNA, when it will multiply, it will duplicate the DNA and one copy will be inserted into each. So here we have a one copy of recombinant DNA. Here also we have a copy of recombinant DNA. Right? So in this manner, the DNA will be passed from one organism into the another. Now in this technology, dear students, when we are using the recombinant DNA technology, we'll be using various tools. Okay, there are various tools that will be utilized that will help us, okay, that will help us to bring about the recombinant DNA technology. And there are three parts, you can say three tools. First is the restriction enzymes. These restriction enzymes are also called as molecular scissors. देखिए कैंची आपने देखी है, जिस तरह से कैंची कपड़े को काटती है, molecular scissors, they cut the DNA. Okay, so wherever we want the DNA should be cut, so they will cut the DNA. Second is the cloning vectors. Okay, where cloning vectors means where that particular inserted DNA will be cloned, and then this recombinant DNA will be introduced into a host, and when this host will multiply, obviously the recombinant DNA will also multiply. So there are three tools of the recombinant DNA technology: the restriction enzymes, the cloning vectors, and the competent host. Now let's talk about the restriction enzymes first. Restriction enzymes. Now basically students, these are the enzymes which cut the DNA at specific positions into fragments. So these enzymes, they will cut the DNA at specific places. Okay, and these enzymes, they belong to a class of enzymes which are called as nucleases, right? These enzymes which cut the DNA at specific place, they are referred to as nucleases. Okay everyone? They are referred to as nucleases. Now in 1963, okay, in 1963, two such enzymes were identified. Okay, two such enzymes were identified, and these two enzymes, they stopped the growth of the bacteriophage in E. coli. तो 1963 में पहले ऐसे 2 एंजाइम्स आइडेंटिफाई हुए थे जो कि बैक्टीरियोफेज की ग्रोथ को रोकते थे ई कोलाई के अंदर. नाउ वन ऑफ दिस एंजाइम्स एडेड द मिथाइल ग्रुप थ्रू डीएनए. एक जो था वो मिथाइल ग्रुप ऐड करता था एंड द सेकेंड यूज टू कट द डीएनए. एक डीएनए के अंदर मिथाइल ग्रुप ऐड करता है, दूसरा डीएनए को स्पेसिफिक पोजीशन से कट करता है. एंड सिंस फ्रॉम दैट टाइम नाउ विथ टोटल 900 restriction enzymes have been identified. So first 2 enzymes were identified in 1963 and from that time onwards till date 900 such restriction enzymes have been identified. And it is very surprising to know that 230 have been identified from the different strains of the bacteria. ठीक है, 900 सच रिस्ट्रिक्शन एंजाइम्स आइडेंटिफाई किए गए हैं जिसमें से 230 एंजाइम्स जो हैं वो बैक्टीरियाज के अंदर मौजूद हैं. दे आर प्रेजेंट इन, दे हैव बीन आइसोलेटेड, दे हैव बीन आइडेंटिफाइड फ्रॉम द डिफरेंट स्टेन्स ऑफ द बैक्टीरिया. नाउ लेट्स लुक एट द फंक्शनिंग ऑफ द मॉलिक्यूलर सीजेस. मॉलिक्यूलर सीजेस, हाउ डज दे एक्चुअली फंक्शन? Okay, now let's see, this is your DNA. Okay, this is your DNA, 5 prime to 3 prime, right? First, we know that DNA is a double helical structure. So it will have 2 helicals. So one is running from 5' to 3 prime and second is running from 3 prime to 5 prime. Now just look at this. There is an enzyme which is named as EcoR1. Okay, this is a restriction enzyme, EcoR1. So it will cut the DNA from this side here and from here. So it will cut the DNA from these 2 specific places. Now if you look at the diagram, you see here in this case the DNA, this particular portion has been removed, and here both they have been removed from each other. So one band is 50, you can say it is here, it is not double helical, here also it is not double helical. So these ends which are now having only single structure, okay, they are only having a single helical. These ends they are referred to as sticky ends. And if you look at the sticky ends, both of them they have the same base pairs. So they are referred to as sticky ends. What do we call them as? Sticky ends. Okay. So restriction enzyme cuts the DNA from the specific places. ये DNA को कुछ specific जगह से cut करता है, and that will lead to the generation of sticky ends. Now how do we name an enzyme? अगर हमने restriction enzymes को discover किया है, how would we name these enzymes? So we are, you can say we are using a very simple method. We are using a very simple convention so that these enzymes can be named properly. Now suppose just now I have discussed with an name of the enzyme EcoR1, right? We have discussed this EcoR1. So this E, it deals with the genus. This letter E has come from the genus. Okay, this letter has come from the genus, right? Because EcoR1, it has been isolated from the, right, E. coli RY13. ठीक है, E. coli RY13 ये इस बैक्टीरिया से आइडेंटिफाई किया गया है, इस बैक्टीरिया से आइसोलेट किया गया है. ठीक है, EcoR1 हैज़ बीन आइडेंटिफाइड फ्रॉम अ स्पीशीज ऑफ प्रोकैरियोटिक सेल. ओके, ये प्रोकैरियोटिक सेल है जिससे आइडेंटिफाई किया गया, एंड द नेम ऑफ दैट इज E. coli RY13. So E means E. Okay, the genus. CO has been derived from the species, and R we can know R has been derived from the strain, and this first is a Roman, this is a Roman number. Okay, this one is a Roman number. Now this Roman number, it indicates the order in which enzymes were isolated. Like suppose if from Eco RY is 13, there are 13 enzymes, or there suppose 10 enzymes have been identified. So this first will tell, this Roman number will tell the order in which this enzyme has been identified. So first letter designates the genus, second and the third letter they designate the species, the fourth letter designates the strain, and the fifth letter it designates It is in Roman number and it designates the order in which this enzyme was obtained or it was isolated. Okay, everyone? I hope this much is clear to everyone. Okay. Now we can categorize the restriction enzymes into two types. हम restriction enzymes को दो categories में divide कर सकते हैं. One is our exonucleases and second is endonucleases. Okay, so we have two kinds of restriction enzymes, right? First is exonuclease and second is endonuclease. Now exonuclease they remove the nucleosides from the ends of the DNA. अब ये जो आपके exonucleases हैं ये DNA के अंदर से nucleotides को remove करते हैं. Okay, they remove the nucleotides. While the endonucleases they cut the DNA at specific positions. आपके exonuclease जो हैं वो DNA को cut करते हैं किसी specific position से. Okay, they cut the DNA at specific positions, right? And they can bind to specific recognition sequences. ये DNA के अंदर कुछ ऐसी sequences present होती हैं जिन्हें वो identify करते हैं. And these sequences they are called specific recognition sequences. Okay, and then they cut the DNA at the specific points in the sugar phosphate backbone. और फिर ये DNA को एक specific location से cut करते हैं. Like suppose let's look at the diagram. Just look at this diagram. Okay, we have a vector DNA and we also have a foreign DNA, right? Now in this case we will be using an enzyme endonuclease, that is EcoR1. Just now we have discussed this enzyme. Okay, E. coli R1, right? Now this is a DNA, vector DNA. Now if you look at this, GAATC, this is a center line. Okay, this is a center line. So they will cut at this position and here at this position. So both the strands of DNA, they will be cut at specific locations. Now once it is cut, now you will have this kind of sticky end. Okay, you will have this kind of sticky end. This portion has been removed. Now same in case of the foreign DNA. Again the DNA has been cut. So you also have a sticky end like this. Okay. Now these two DNA fragments which are having the same sticky ends. Both these DNA, they are having the same sticky ends. So now this foreign DNA will be joined with the vector DNA. And this will form a recombinant DNA. And this joining is done by DNA ligase. ये जो दो DNA की joining है, two different types of DNA, they will be joined by the DNA ligase. So there is an enzyme DNA ligase which will join the two sticky ends. That is, it will join the sticky ends of the vector DNA with that of the foreign DNA. And this particular portion is referred to as is having the recombinant DNA. ठीक है, इस पोर्शन के अंदर जो DNA आपका मौजूद है उसे अब हम recombinant DNA कहेंगे because it contains two different types of DNA. One from the vector DNA, another from the foreign DNA. Okay? Okay, everyone? Right. Now coming to the enzyme, the first restriction endonuclease which was identified was Heinz second, and it cuts the DNA at specific or at particular points by recognizing a specific sequence, and that sequence has 6 base pairs. Okay, that sequence has 6 base pairs. Now this sequence is called a recognition sequence. Okay, this sequence is called a recognition sequence, right? Now your restriction enzymes, dear students, they can also recognize certain sequences which are called palindromic nucleotide sequences. They are referred to as palindromic nucleotide sequences. Means the sequence or the base pairs that read the same on both the strands, whether you read it from the 5' to 3' or from 3' to 5' direction. Let's take a very simple example. Suppose if I use a word delad, okay, now read it from here, it will be delad, and if you read it from this side, it will also be delad, right? So you can read from both the sides, but the word will remain same. Similarly, there are certain sequences in the DNA. You can read it from the 5' or the 3'. They will remain same. And those sequences, they are called as palindromic nucleotide sequences. What do we call these, students? Palindromic nucleotide sequences. Okay? I hope this is understood. Is this much clear to everyone? Right. Like as you can see the example here also. We can have a little example here. Like see this sequence. GAATC. I am reading from the prime to 3'. Now read it from here. GAATTC. So such sequences, they are referred to as palindromic nucleotide sequences. Okay? Understood? The sequences which mean same, which are read same from both sides, those sequences are called palindromic sequences. So they can be read in both directions. Okay, you can read it from the left, you can read it from the right side or the left side. They will remain same. There is no change in them. Okay, so restriction enzyme, they cut the DNA a little away from the center of palindromic sequences. तो जहाँ पर आपकी palindromic sequences है, उसका जो center है, उससे a little away from the center. So they cut the DNA from a little away from it. Suppose this is a center. ये आपकी sequence का center है. Okay, this is a palindromic nucleotide sequence, means the sequence which reads same from both the sides. So आपका जो restriction enzyme है, वो सेंटर से कट नहीं करेगा, बस उससे थोड़ा सा दूर से कट करता है. राइट? Now because it is cutting it from a little away from the center, this is because of it sticky ends are created. Sticky end means the hanging stretches are formed. Single stretches are formed. जैसे कि आपको यहाँ दिख रहा है, ये single stretch है, ये भी single stretch है. So these ends, they are referred to as sticky ends, and these two sticky ends, they can be joined together by the DNA ligase. Right? These can be joined by the DNA ligase by forming the complementary hydrogen bonds with the complementary base pairs. Right? Now this, if you look at these two DNA, the vector DNA and the foreign DNA, both of them, they are having the same kind of sticky ends. Both of them, they have same kind of sticky ends, and thus the DNA ligase is able to join them. Right? So for today, you will be making notes of this topic, and you will read the page numbers 193 to 197. And dear students, if there is any doubt, anything that you want to know, you should send your doubts or queries to my personal number. Right? So that's all for today. Bye everyone.